So we have heard for many years just, like kind of conflicting perceptions or messages. Some, government agencies saying the water's no problem, even though the contaminated sediment is at the bottom of the river. Water is totally safe. But lots of community folks always having questions and curiosity about if that's really true, and more than anything just wanting to know like what actually is in the water, and are there any things that are actually untested for, that the city of Portland doesn't actually test for, or whoever doesn't test for, that might be in the water. And so we had learned about some technology for testing unregulated contaminants in Europe, and got in touch with OSU to see if they had heard anything about that type of technology for water sampling. And it turns out that they have access to that technology. And then we teamed up together a couple of years ago and decided to do some community-based research, and that's how it got. That's how it went. And we had several meetings with community folks to identify kind of the best places to sample, and sort of the scope of the area, the geographic area to cover, and sort of reached consensus amongst the researchers and community about how to proceed. So that's anything, Laura or Jamarian, that you want to add?
Yeah, I would add that around the same time we started working with you folks, we also did some PCB passive air sampling that showed us that PCBs were indeed airborne, which EPA always denied. However, we subsequently learned that they were doing their own tests. So they knew about it, but this is my interpretation. They didn't want us to know about it. And I've also learned from Dr. Carpenter that this is not, publicly stated, that PCBs move in the water too. There are lots of ways they can creep into our bodies. So they don't always stay fixed in the sediment. And then with your sampling, it was just really exciting to maybe see what's actually floating around in the water. So, preliminary results have been very meaningful. Jamarian?
Hello, can you guys hear me now? I had to exit and then rejoin.
Sorry, but yes, we can hear you.
Okay, good. So one thing I just wanted to chime in about is just their methods of testing, how they differentiate from the traditional city methods when testing the water, and being able to test such a wide range versus, one at a time, at the different points that they're at, because at different points we have different contaminants and toxins that are in the water. So I just wanted to highlight that one point. And I hope you guys heard me because I'm in traffic now. Sorry.
No, we were able to hear you. Thank you so much. All right. So with that, this meeting was organized with Portland Harbor Community Coalition to sort of go over some of the results that have been reported out. Once Manuel starts talking, he's going to go through pretty much everything that we have in that report. I will also drop a link in the chat to the website that has more of this information, as well as a video and some flyers. But the goal was just sort of have Manuel talk through some of this, and then we've left a lot of time. I think, Manuel, you're planning on talking for 15 or so minutes, but we wanted to leave a lot of time for people to ask questions, just have a conversation about the results and what we're finding, where we might want to go from there. So with that, Manuel, take it away.
Excellent. Thank you so much, Diana. Can everyone see the screen and full screen mode?
Yes, we can.
Excellent. Thank you. So first of all, thank you, all of you. Thank you to the Portland Harbor Community Coalition for organizing this forum. And I'm happy to be here with you all today, representing this large project that, as Diana has mentioned and others, it was started actually by the Portland Harbor Community Coalition when they first reached out to Diana asking for support for this expanded water monitoring capabilities. And also, it was very good time for us to have our new PhD student at that time, Chloe Fender, jumping into this project that was pretty much her baby from the very beginning, and she successfully completed before her graduation earlier this past year. So I would like to thank both Diana and Chloe for your work on this project, and to the Portland Harbor Community Coalition for the opportunity to present some of our results here today. So without more details, because has been already explained how this collaboration study started, it was a community initiated project, and there were some fundings at OSU that we leveraged for getting started this project. However, those funds were not enough for the completion of the project, so we had to find additional sources of funding through my laboratory to be able to complete all the analysis. But it was just the initiative from the Portland Harbor Community Coalition who made this possible. So thank you all for that. Why we were interested in the Portland Harbor? As it had been mentioned, there is a little bit or a large gap of knowledge regarding the contaminants that are monitored in regular basis in the river. We know that has been like common knowledge, some of the main contaminants that are made present in the sediments, and we know that some of those contaminants are related to pHs, industrial chemicals, metals, pesticides, and dioxins. Some studies have also focused on some of those contaminants in the water column. But this is a very little amount of compounds if we want to compare with the vast amount of contaminants that we have in the environment nowadays. Can be hundreds of thousands of compounds. So why the EPA decided to focus on these compounds? These are compounds that have been well reported with a well-known toxicity that we know that were released, and they were found in larger concentrations in the area. So that's why they prioritize these compounds. And we are not saying that they did not a good job. Well, we want to expand the number of compounds for further assessments based on the new technologies that we have now, to bring their attention potentially new compounds that they should include in the regular monitoring capabilities that they may have now. That was the whole purpose of this, just to leverage on efforts that are currently out there. So with the help of the Environmental Health Science Center, Diana, her team, Chloe and I helped also with the development of this infographic, just to give a better understanding of why we decided to implement non-targeted analysis into this study. Traditionally, with a targeted approach, we are limited to the number of compounds that we can analyze depending on the number of standards that we have in the lab. I have a standard for ibuprofen, and I can detect or quantify ibuprofen in water. So that is very limiting, because we don't have enough funds, time, resources to buy analytical standards for all the compounds that may are in the environment. So we can make a better use of information that has been acquired in other labs, and they can share with us. It's open access, and they say, this is how it looks when you analyze ibuprofen in your system. And I'm going to share this data with you so you can do a suspect screening in your lab. And in that way, we were able to tentatively identify a larger amount of compounds. However, we can even further expand the search by going further and trying to get a more comprehensive understanding of the overall composition of the sample by using a combination of targeted, suspect, and non-targeted analysis. And that is what we implemented in this study, especially a combination of the last two. This allowed us to monitor hundreds of compounds versus a few dozens that are currently monitored by the EPA and others. As Casey referenced before, we met regularly, and it was a lot of fun, actually. I remember one event that we met in person in the river, and everything was great except for my car that got broke in the bridge just before getting there, and I had to get a tow to go back home. Thanks God that my wife was able to pick us up. But yeah, it was very nice evening, and I really enjoyed meeting with you all. And I learned at that time more about your PCB's air monitoring campaign. It was very informative. Enjoy learning from the DEQ and others about this project and other topics. So with Portland Harbor Community Coalition feedback and the community feedback, we were able to select 6 sites that were feasible to collect samples from that met our criteria and that we were able to monitor monthly for a 6-month period. This was a large effort. I want to mention that Chloe decided even to become a captain, and she navigated the river with others. I only joined for the first couple of times while we were familiar with the river, but she took over, and we were able to monitor 6 times there for water contaminants. That means that we pretty much used a very sophisticated technique that implied throwing a bucket from the boat and collecting water, of course, with a lot of standards, protocols for rinsing our bucket, cleaning buckets, taking measurements and taking samples in the prepared bottles. We want to make sure that we minimize the introduction of contaminants, right? So all of the methods were validated in my lab, and we were able to do this in a timely manner from May 2022 that we got the funds. We started the sampling in June, and we were sampling until November. And all the data, all the samples were processed and analyzed during 2023. And here we have some pictures of Chloe. She's here taking some measurements with a pH meter with the water. She's dumping the water in the bottle. The bottles get labeled. This is Miranda Jackson, another PhD student in my lab. And all of that for what? All of that to go back to the lab and do a lot of fancy extractions, analysis, data processing, and put all of this together into big data sets that are going to tell us about different contaminants that we were able to detect or tentatively identified. In this case, we put together all the data for the different months and the different locations, and that gave us around 900 organic contaminants. Please note that the fact that we have 900 organic chemicals tentatively identified does not confirm their identity, because we don't have 900 analytical standards in our lab. So these are compounds that, depending on their abundance, potential toxicity, the frequency of their occurrence, we will further prioritize. And I will talk about the next steps. But these give us a very good first understanding of which are the main contaminant or classes that are present in the river, and also tell us about which contaminants should be prioritized for further testing. Just to give you an idea, of course, we found some PFAS, we found a significant amount of food additives, we found transformation products, that these are just a result of physical, chemical, biological transformation of other parent compounds, could be pesticides or pharmaceuticals, etc. We also found plastic-related chemicals, household pesticides. We found a significant amount of personal and household products, personal care and household products, as well as industrial products and pharmaceuticals. If you see, pharmaceuticals compose a significant amount of the 933 organic contaminants tentatively identified. And then, this is not surprise, we have a large amount of compounds that are coming from plants, microorganisms that are naturally occurring in environment, and that's good. That's part of the environment. We need to be happy to find those natural products. There are a few exceptions of natural products that are not very pleasant or not and pose a significant risk. It's the case of, for example, algal toxins. But please know that most of the natural products are not toxic in the environmental concentrations. And then there is a significant amount of compounds that we were not able to classify. And this is not because our libraries were not able to find them. This is because probably they are used in different practices, and we were not able to assign a main usage. other ones that have been found, but they have not been easily linked to an usage. So this may occur naturally or not, but we cannot tell you where they are coming from at this time. Let's keep moving. One of the most interesting discoveries in our study was related to the timing of the presence of the contaminants. And when we try to group the data based on how the data distribute, if it's distributed more, the differences are coming more from the different locations versus a different time of the year, we clearly see that the samples that were collected different times of the year are more different than the samples when we compare between different locations. That kind of makes sense, right? Because overall, the river changes during the year. We know that the flow is different and during different times of the year. We know that the rain has a different impact on the dilution of some of the compounds that may are present in the river. So this was not a surprise. As you can see in this bar graph, we found a much larger number of compounds during July and August compared with the month of September. It seems that in September, we were able to find a less number of compounds. That doesn't mean that some of the compounds that we found are not important, and we will go into that in a second. Let's just take a few minutes to highlight some of the chemicals that we were able to identify. Some of the compounds are related to pharmaceuticals, as I have mentioned before, and in the case of irbesartan, this is a very common drug that is being used to treat hypertension. So it's not surprising that we could find traces of this pharmaceutical in the water bodies. At the same time, it's no surprise that here in Oregon, we are big users of antihistaminic drugs, and fexofenadine is one of those active ingredients that is contained in many antihistaminics. Another compound that was found in the water body was methylbenzotriazole, which is an industrial derivative. This is an industrial chemical that is being used in very different applications. I won't get into the details, but this is a common contaminant. And then here I put two examples of natural products that are coming, in this case, from plants. Altamisic acid or altamisic acid. This is a naturally occurring product. Is coming from. This is an invasive weed here in Oregon, Ambrosia tenuifolia, and it's not surprising that some of the natural products produced from these plants are also present in the river. And finally, also highlight a compound that is also derivative from a plant. In this case, this comes from different places, but mostly from Uncaria rhynchophila. And this is actually a plant that is being used in medicine, in Chinese medicine, also to treat hypertension. So I don't know if this is just coming from natural plants in the environment, or is as a derivative from some of the natural products or supplements that people is taking. All of this to say that the diversity of contaminants or compounds, because in some cases are not toxic contaminants, are just compounds that we are finding, is very diverse, is large. We have a large diversity of compounds. And with that, looking into the sources, we should take a moment to reflect into the different ways that a contaminant can get into the environment, right? We have contaminants that are coming from the industrial wastewater effluent, and this is something that the Portland Harbor has seen, unfortunately, a lot of releases, and nowadays it's very regulated, but there is still some heavily industrial activity in the area. So some of those contaminants are likely coming from there. We also have agricultural runoff, and this is something that we are getting better at identifying, reducing nitrates and in water bodies, reducing pesticides leaching, and this is something that is more regulated nowadays than it was in the past, but we still see many pesticides reaching the water bodies and the surface water bodies and groundwater. We have contaminants coming from rat runoff. We drive cars. We are contaminating the environment in daily basis, air pollution, water pollution. Nowadays, we know more about contaminants that are coming from the tires itself, and some of the chemicals that are in the tires get into the surface waters as well. We have contaminants coming from landfills, and of course, contaminants coming from our households, just products that we use at home, as well as pharmaceuticals. As we excrete, some of those pharmaceuticals, they ended up also in the wastewater effluent. So all of this is contributing to the health of our water bodies, the ecosystem. And in the particular case of the Poland Harbor, we have a comparison between site 5 and site 6, and it was very interesting that we could find that site 6, most of the time, it was different in the composition, the number of compounds and the classes of compounds that we found, and the abundance of those compounds, it was significantly different than the other sites. And we hypothesized that related to the fact that site 6 is downstream to the water treatment plant, a large wastewater treatment plant. So maybe some of the contaminants that have not been treated or retained or degraded are coming out at that point. And that is why we may find more of those in the intersection with the Columbia River. That is also something to consider. We could have some contaminants that are coming from the Columbia River, but this is a much larger body of water, as we all know, and probably more diluted in some cases. So we hypothesize that the higher abundance of contaminants in Site 6 is likely more due to the presence of the water treatment plant. I will mention in our next steps, but my lab is also interested in wastewater effluents. We have a couple of projects. One of them is an EPA-funded project. It's a 3 years award that we are currently working on, and we are specifically looking into organic contaminants coming from different treatment plants, in particular 40 treatment plants, wastewater treatment plants across the US. This is a large project in collaboration with engineering here at OSU and microbiology. And we are doing an effort to try to identify better practices to reduce amount of contaminants in wastewater. So that is one thing that we are also interested. Another project that I would like to mention later is related to the Columbia River Basin, and I will focus more in a following slide. Just to finalize with our data, the data that you saw in the report that was shared with the community last year, at the end of 2024, thanks to the large effort of Diana and her team, Chloe, they worked together to get this report back. We were able to share with you all some of our exciting results also from the zebrafish study. And here is what I want to say. This is not all bad news. When we tested the water with the zebrafish, even when the water was concentrated, we could not find high toxicity. The fish were like not having very hard time. We saw some responses, and those responses were higher when we compare September and November, were higher in the month of November. And were higher in site 1, site 3, and site 5. So for some reason, when we expose the zebrafish, that little tiny fish that is very, very young, the embryo stage, when we expose them to water, even concentrated water, we saw a slight response, but was not very different than the controls. So that's good news. I don't want to let you with the feeling like our water bodies are contaminated with 1,000 contaminants that they pose a risk. Risk is different. We can tell you what we can find, but in order to be toxic or have a negative effect, we need to consider other factors. We need to consider our exposure. We need to consider the concentration of those contaminants, and all of those factors were not evaluated in this study. Okay, so please bear with me during this presentation, because I don't want to sound alarming when I say we identified 1,000 compounds. We need to consider the compounds that are important for us to further test. So this is what we found. These are compounds that have been found in other water bodies. Actually, our suspect list, that information that we use at the beginning to compare with, is not coming from, it's coming from very curated, specific sources. One of them, the largest source, is coming from Europe, is a Norman list that is just an effort, European effort, to put together contaminants that have been found, in this case, mainly in water bodies, in different rivers and different lakes and different drinking water sources. and we took all of that information. So our sources are curated. It's good quality sources. So we had a good thing to compare with. What did we find? So we found that, as I say later, we didn't see any differences in weather samples, not major differences due to the Superfund site. That is important, and there is a highlight from our study. Main difference were coming from the different times of the year. Also to highlight that we were not looking into metals or PCBs or other legacy contaminants. We were mostly interested in contaminants that are in the water body. That means that they are highly polar or that they are moderately polar. The compounds that are very hydrophobic usually get retained into the sediments. They're more difficult to analyze, and there are other methods to do that. So as I said before, there is a trend. The trend is likely to be related with the time of the year. And yes, we know that different practices happen at different times of the year. For example, agricultural practices. There are some months of the year that we have more input from agricultural runoff because people is spraying their fields. And that is not surprise. And yes, good news that the chemicals, the organic contaminants we identified were not tested individually to the zebrafish in higher doses to see, or understand better the mechanisms of action of those contaminants. Rather than that, we tested the whole water, the whole thing, how the whole thing will affect fish in the earliest stage when it's very, very sensitive to this water. And we found very little effect. Okay, we saw low toxicity in the zebrafish, and this is good news. Okay, next steps. I want to leave you with some good news, and the good news is like we keep working on the compounds that we found. This is not just something that we put in a publication that we recently did and forget about it. We keep looking into the compounds that we decide based on our data that we selected for further prioritization. That means that in our future studies, we will keep looking for those specific compounds to see if we keep finding them, and also to learn more about their concentration. The concentration was not an important factor in this study. This was more a discovery study, but now we will be looking more into concentrations of the prioritized compounds, the frequency, and the relative abundance compared with different sites. And I would like also to highlight that our university was recently awarded with an EPA Columbia River Basin project. And this is a 5-year project. Diana is part of this effort together with other 5 co-investigators here at OSU. I have the privilege to lead this work, and we are just getting started. It was recently funded, and the kickoff meeting happened earlier in 2025. We just recently met with our advisory board, and it's very exciting news because we are not only going to corroborate some of the results that we found in the lower Willamette River, but here in this project, we will have the opportunity, as I want to just mention in the next slide, to characterize spatiotemporal trends of organic contaminants for 1 year. We are going to use passive sampling devices. That means that we are going to dispose with the help of the Anderson lab here at the EMT department at OSU. We are going to install passive sampling devices every month and collect them every month for a full year to get a full characterization of almost a 200-mile section of the river, of the lower Columbia River Basin, to have a better understanding of the contaminants that are bioaccumulative, contaminants that potentially get absorbed into the fish, contaminants that may are potential risks also for the sea lions that are eating those fish, contaminants that may are impactful, a negative impact in the people who is eating those fish. So this is an important project for us. We will also be able to match the results from this project with the Columbia River Basin. In this project, we are also going to use novel methods for toxicity testing, like the zebrafish, and also human cell lines to see direct effects in the human population related to the contaminants that we prioritize. And we will also be studying the different sources of the contaminants. Through all of that, we will stay engaged with community, and we establish a very strong advisory board. And I would like to mention that one of our sub-awardees is the Columbia River Keeper that is going to work closely with us during the 5 years. So all of these, thank you to the the great support that we have from the Poland Harbor Community Coalition has inspired a lot of this work. So thank you all for your support through the years. And this is just a final slide to thank you all. Thank you, Chloe. Thank you, Diana, and our funding agencies, our happy instrument that is always there for us. And with that, I will be happy to take any questions if you have.
Thank you, Manuel. If you don't mind, well, actually, Laura, if you had a question, and I see.
Yeah. Manuel, about the wastewater treatment plant, is that effluent coming from the plant on a regular basis, or are you talking about the spill that comes when there are heavy rains and you know, we're told not to go into the river?
Yeah, thank you, Laura. That's a good question. So each waste treatment plant operates differently, right? Each of them has their own treatment processes, their different inputs, outputs. So in this case, we have a very good water treatment plant here. It's not perfect, it's pretty good. And the water, after being treated, is being released into a huge pond where it keeps being further treated. But at some point, that water has to be released, cannot be contained there forever. So that at some point, the water is being released into the lower Willamette River and then the Columbia River. So what we are seeing is that even with their fancy treatment capabilities, there are many contaminants that are not being treated.
Yeah.
Because they cannot get like retained to the different filters that they have. They don't get degraded because they don't have enough biological capability in the bioreactors or whatever. So we are testing now in the other EPA project. We are testing with folks 40 different water treatment facilities with different treatment processes to see which are the best practices that we could implement depending on the kind of water that we get for different places. So I believe that those results are going to be impactful at the national level and that hopefully in the next few years we can see some implementation.
Great. Thank you.
Thank you, Laura.
We did have a question in the chat as well, Manuel. So Ashley, I can ask this for you, but you can also jump in and correct any of this. But Ashley was asking if your lab is one of the technical partners at the Willamette Partnership Northwest Environmental Justice Center.
Oh, thank you, Diana. Actually, I am not aware of that. So I will be very grateful if you will share more information about that. And I offer my lab and the capabilities that we have here as much as we can. We are very busy with projects, research projects for our students and our current collaborators, but we are always open to new ideas and new potential collaborations.
Awesome. Thank you. I appreciate it. It's one of the U.S. Environmental Protection Agency's technical centers that they funded in our Region 10. And so I'm not really sure how the selection process went, but yeah, it might be worth reaching out to them if there's capacity with your students at all.
Thank you so much. Maybe something to consider in the next few years. Laura.
Yeah, I have another question about the fact that the river is tidal. The Willamette, I've heard that actually the Columbia is tidal up to as far as the Bonneville Power Plant Administration, the dam. So the Willamette is also tidal. I'm not quite, I don't know quite sure, I'm not sure how far up it goes, but that must have some impact on what you're looking at too. Did you factor that in, I wonder?
Excellent question, Laura. Actually, I don't think that we factored the tides. It's something that we will have a better understanding now with our new sampling design in the Columbia River Basin. But we didn't factor. We just went there for different months of the year. And I believe Chloe is here to correct me if I'm wrong. I think that she looked for some correlations with the precipitations, the flow, but I don't know if we specifically try to factor the tide. Chloe, please feel free to chime in.
Yeah, and if you can also mention, I think you were sampling around the same times of the day each time, if you want to mention that, Chloe.
Mhm.
Yeah, definitely. So we did sample at each time of the day, or like around the same time each time we went out and sampled. But often when the river level was higher and there was more like ponding from the higher water and from the tides coming in, we saw a lower concentration or a lower number of chemicals detected. So in November, we had a lower number of chemicals detected overall. Same with June, where there was a lot of runoff. And then we saw a lower number of chemicals detected when the river levels were higher. But the tides are pretty hard to sort of control for because they're moving both directions. And so we did treat the system more like a river in our understanding, but it's definitely a unique river compared to maybe like a small river or even the Willamette, like, upstream from Eugene area. And my understanding is that it's tidal all the way up to Willamette Falls, and then it's no longer tidal.
Thank you.
Mhm.
I see a couple of questions here.
Jamarian asked, what was the most toxic and most abundant finding of the 933 substances found?
Mhm. That is a very good question. As I would like to highlight again, we did not test for individual toxicity of any of the contaminants that we found. So this will be part of the next steps. Now that we have prioritized for the contaminants that we are interested the most, we will go ahead and keep doing this testing. For me, it's important when we are talking about the toxicity, it's important, of course, to consider the exposure. This is a contaminant just because it's there, yes, but it's not means that this has a negative impact. We need to consider the bioavailability of that compound, how that compound gets into our system, if it can get in our system. So we will need to consider all of that. I am not a risk assessment expert. I am more like in the analytical chemistry environmental side. So I work with molecular toxicologists like Dr. Robin Tangway and her lab and others, like for example, Diana Rohlman, to translate the results. And I would like to say that this is part of our next steps. And that's why in our new EPA project, we are making a huge emphasis on novel approaches or novel methodologies for assessing toxicity. And this will involve not only testing the highlight compound that we say, okay, this is a very toxic pesticide that we found. We want to see how those compounds behave in the environment together with other ones that they are found at the same time. So for example, if this specific pesticide is usually found with this other, in this case, pharmaceutical, how both two in the in the sense that how would they interact in our system if we were exposed to both of them at the same time. In the case of drinking water, for example, we don't drink one contaminant at the same time at each time. We are drinking the water. So Diana, please.
I was just going to go back to Jamarian's question. Those are the types of answers or questions we want to be able to answer.
Mm-hmm.
You have to do studies like this to even figure out what is there. And a lot of those studies haven't been done. So I would say the biggest impact of this study has been being able to go out, collect the samples, and just identify what is there. And like Manuel said, I know that 933 number sounds really concerning. The number of chemicals is not actually something that denotes if it's going to be toxic or bad, especially as Manuel mentioned, a lot of those are natural. I know Chloe has also done some work, and so I don't want to put you on the spot, Chloe, but Chloe has done a lot of work looking at what's been found in other urban rivers. And so I believe, Chloe, you said this was actually pretty common with what's been found in other urban rivers throughout the US. So hopefully that context also helps. Of course, we're not saying that just because it's common, that means it's okay. We're not saying that. We're also not saying that means it's bad. Really what we're saying is this study was able to identify chemicals that have been found in other rivers. And that allows us then to do future work to say, of these chemicals and contaminants that have been found, should we be worried about them? And that is really the next step. We don't have enough information to fully answer that. So Ashley wrote, is the study essentially creating a reference collection of chemicals present in the river and study area? To an extent, yeah. I mean, obviously there's going to be other sampling techniques that Manuel talked about that would look for other chemicals, right? Like we didn't sample the sediment, and we know that there are specific contaminants that are already in the sediment. So Manuel's study is going to focus on the water column. So yes, Ashley, you absolutely summarized that.
Thank you so much, Diana. I see a couple of very good questions here in the chat. I will start. I think the next one will be when we speak of the fate of contaminants. Is that right? Yeah. What involve the Columbia County EPA rates? So it's a Cass, would you mind to extend a little bit on this question to clarify the point that you would like to make?
Ask, are you able to unmute and provide a little more information?
Yes, hello. We are concerned about the tidal influences and the contaminants that wash back and forth because we drink water that's even below our own sewage excretion, which is into the Columbia, right where the Willamette has split into the Multnomah Channel. So the Centers for Disease Control and EPA have county by county ratings for cancer incidence. And I found it very, very odd that Columbia County should be number 1 and sometimes number 2 county for cancer incidence. I know that correlation is not causation, but it's an interesting coincidence to be at the receiving end of this very large flush from three or four states and a province.
Yeah, Casa, this is very, very interesting. Thank you very much for sharing that. And I mentioned here our interest in wastewater, our interest in surface water from this project, but we are also actually studying drinking water. And we have another funded study in my lab where we are currently recruiting participants, and we have recruited also with Diana's help and actually OHSU. We have recruited around 450 participants across different locations in Oregon, and we are also monitoring for drinking water contaminants. It's a similar scope to this. We are in the discovery phase, and then prioritization of contaminants in drinking water based on occurrence, abundance, and toxicity, and we will be reporting on that in the years to come. And this is an ongoing research on my lab. So please be aware that we are currently studying. We will not be able to tell you if the cancer that you are seeing, higher incidence of cancer in the area is due to this, but we are going to make efforts to address contaminants that are found in drinking water and how those contaminants vary in different locations and different times of the year in Oregon.
Yeah, also, Cass, I'm looking for it right now, but I did some work with Beyond Toxics, so down in West Eugene, where there's also a higher cancer rate. So I can drop into the chat a link where we just sort of talked about like the difficulties of connecting environmental exposures to cancer, which is obviously what everyone's very interested in. One of the biggest difficulties is that typically when we're looking at environmental exposures, we don't actually see increases in cancer for 10 to 20 years, which makes it difficult from a scientific standpoint to sort of prove that. So I can put some of that information in if you're interested. I'm also happy to chat with you more about sort of the scientific difficulties of looking at those connections.
Yeah, thank you, Diana. And I think one of the very interesting factors of this study that we have done, that we are currently doing with OHSU, is that we are leveraging in our recruitment from the HOPES, the Healthy Oregon Project cohort that they have. So that means that we don't have that data yet, but we are monitoring water from households from people who have been tested for cancer. So at some point, this data will be accessible. And that is something that we want to highlight. We try to make our data accessible for other scientists, epidemiologists, other biologists, other toxicologists that in the future may keep working on these important questions.
Yeah, this is staying with in this study.
Yes. Next question.
We have another question, which was from Clancy. What are the contaminants that have been prioritized for the next phase of study?
Great. In order to answer that question, I would like maybe to share my screen again, if that's okay. Okay. And you all see this again?
Yes.
Okay, so this is another slide that we put together from our study. From those 930 compounds, after a lot of work, we were able to highlight a few of them. In this case, 128 compounds. And this was done based on the different usages of the compounds and also based on what we knew about those compounds. Compounds that have been already tested in other studies, compounds that have been shown to have toxicity in other studies were included here, but also compounds that are coming from classes that we want to highlight at this time because, for example, pharmaceuticals is a large component. And in some cases, I mean, all the pharmaceuticals that are released have been tested for toxicity, but that doesn't mean that someone who has the prescription drug, and is for that person. That doesn't mean that we all need to have that contaminant in our water, right? So contaminants that have been tested for toxicity doesn't mean that it's good for everyone. So in this case, we highlighted pharmaceuticals because they represent a large amount of the contaminants that we found. And this is one of the reasons why in the wastewater treatment project, we are focusing specifically into antibiotics, because there is a lot of antibiotics in the wastewater that are not accounted for. We also highlighted a group of 29 pesticides, common compounds that are applied in Oregon for further monitoring. And again, we want to know about the risk of being exposed to the water itself, the combination of those compounds. In order to do that, there's a lot of testing to do. We need to test the individual compounds. We need to test the compounds in combination to make relevant mixtures. So this is not something that I can give you an answer, quickly answer here to tell you which of these 128 compounds is more important. That's why we highlighted this section, and we will further elaborate into the contaminants that we found, combining with information of our follow-up studies.
The other thing to know here, and Manuel talked about this sort of first phase of work, which is incredible because it's able to identify all of these different types of potential contaminants. We're not able to get the actual concentrations. So I know, Manuel, that's also something that you've been interested in, is identifying some of these compounds of interest based on how frequently or how often they're being found, and then actually trying to do additional work to get concentrations in the water, which then helps us get to that next step of, are they toxic at these? How are people being exposed? So it is probably one of the most frustrating things that I have to always acknowledge with science. It is slow. It is stepwise, but this is the best way we know to try and answer these pretty, it's an easy question to ask. It is so complicated to answer, which I know is one of the most frustrating responses to get. But this is one of the best ways we know how to start. Find everything we can and then narrow it down and get more and more information at each step.
Thank you, Diana. Yeah, just for a little bit of context, imagine that each of those compounds, if we wanted to give you an idea of the concentration, we will need to buy the analytical standard. Each of those analytical standards, if it's available, is going to be somewhere between $200 to $500. So just make numbers. It goes to very, very expensive, very fast. So we need to be very selective with the compounds that we decide to further monitor.
Yeah, it's also a time. It's trying to narrow in on what is most important to get those answers quickly. You also have a question from Charlotte on, can you further define what a plasticizer is?
Yeah, thank you. So plasticizers, we refer to, for example, compounds that are added to the plastic to add some properties, right? For example, some of the phthalates, compounds that are added to add flexibility to the plastic. So things that end up
not break.
Yeah. So that's what we mean with plasticizers, compounds that are used in plastic. Sometimes the plastic itself is coated with additional things. And those are the compounds that we are exposed the most. The plastic is a little bit harsh compared with the other chemicals that are added afterwards. And those are the ones that are, for example, released. When we put a Tupperware in the microwave, we get more exposed to the plasticizers, the chemicals that are added to the plastic itself.
Yeah, I will say they do serve a pretty useful purpose. One of the other times we'll hear about plasticizers a lot, there's been studies, folks that have been in hospital with IVs will have higher levels of plasticizers. But as you can imagine, those IV lines need to be really flexible. So that is why they were developed. You also have a question from Laura, if you can give redefine a transformation product.
Yeah, thank you, Laura. So imagine the case of, we all know about Roundup, right? Roundup is a glyphosate. That is the active ingredient that it gets applied into the fields, but that compound doesn't stay like that for very long. Actually, in a question of a few days, it's transformed into something that is called AMPA. So that is a transformation product. How that can happen? If it's mediated by a molecular organism like a bacteria, we call it a biological degradation product. But it can be also formed by physical or chemical processes, like just being in the water in combination with the water. It can create, based on hydrolysis, can break down in other things. Or the light transform and degrade the compounds as well. So those are transformation products. Why is that important? Sorry, Diana, I just
Oh,
wanted
no,
to
I was going to get to that. Like, it's really awesome that you're able to see these products, because in the past, a lot of times people just looked at the original chemical and they'd say, oh, is it there or is it not? And over time, it goes away, except it's not really going away. It's just breaking down into these transformation products. So that's a really cool thing that this method is able to identify those.
Yeah, and why is that important? Because in some cases, the transformation product can be more toxic than the parent compound. And that has been proved for many compounds. In our lab, we are also very interested in pHs. Actually, our department is one of the main sources for pHs research across the US. And we are interested in transformation products toxicity, because in some cases has been shown to be higher than the parent compound. I'm talking about the polyaromatic hydrocarbons. Okay.
Are there
No, this is a lot. I just want to take a moment to acknowledge and to thank you all for bearing with me with so much information.
Yeah, I've been trying to put resources in the chat as well as they come up. We're happy to stay on. I also know it's been an hour and folks have busy days. So Jamarian asked, it'd be interesting to see how many initial products converted to transformation products at the 933. Yes. I will note, I don't do this work, but there are folks that solely study how chemicals are broken down and the metabolites and all of those changes that go through. So we've got some partners up at Pacific Northwest National Labs that do a lot of that work as well.
Yeah, and as you all can imagine, it gets complicated very fast, right? One single compound can be degraded or transformed into dozens of compounds. So we need to be a little bit selective and trying to find things that are relevant. But yeah, we are looking for many more things that we used to with the traditional approaches. So we are very happy that we are making a difference, but still it's not perfect. We have some compromise here.
So except we're happy to stay on if people would like to continue asking questions. I'm going to put my contact information in the chat. Y'all are welcome to send questions to me, but then I will be sending them to Manuel, Portland Harbor Community Coalition. So we get those information. We've been talking with Cassie and Jamarian about putting together sort of a frequently asked questions page. So I know there's been some other questions. So feel free, if you remember it, if you're like me, you wake up at 1 a.m. and you're like, oh, I should have asked that question. You can send the email and we'll get back to you.
I had a question in regards to the follow-ups that were mentioned, in regards to the collaboration with Columbia Riverkeeper. How would we best be able to follow that work? Is it like through EPA, which might not be a great way to subscribe to right now? Is it through Oregon State, or like where do we go?
You can email myself and Manuel.
Okay.
We're still really early in that, but some of the questions we're going to be having for folks, are what is the best way to get information out? So Columbia Riverkeepers has a pretty well-established social media presence, newsletters, etc. We've talked about infographics, story maps, other sort of ways to get all of that information into one place. But yeah, you are welcome to reach out to us.
Yeah, and then like the follow-up questions that it might be just be my limited understanding of the river keepers, but like I understand that some of them have like legal counsel or like staff in-house. And so I was just kind of wondering if that changes your study for like if they plan to use it for any sort of like litigation, or if any of the people that they work with do. Like I know they work with the tribes in Washington, for instance. So I was just curious about like if that's something that you all have to think about in regards to the study design.
So this study, and well, you're welcome to jump in. This study was written by us here at Oregon State. So they're a community partner, but the research design is going to be led by OSU researchers. What we have committed to do, and we do this with all of our projects, independent of who we're partnering with, is we try to make sure that our information is available to communities that want to use it. And then we do make our data publicly available. At that point, once it is publicly available, we have we have limited control over how people choose to use it. But the data is owned by the OSU team at this point. Well, we haven't collected data yet, but that is how it would be working moving forward. Does that answer your question, Ashley?
Yeah, until it goes to like a public database or something, unless that's also owned by OSU, is kind of what I'm hearing.
Yeah, Manuel, is there a public database for non-target analysis? One of the things we're running into is some of the data that we collect, there are not existing public use repositories. We do have one repository here at Oregon State. It's our Superfund data repository. And so we'll make data available through that if no other existing repositories exist.
Yeah, that's a good question. There is a few cases, like for example, the pesticide stewardship. They put together a map, interactive map, to see different concentrations for specific pesticides in different rivers or streams in the area. But that is just for the few compounds that they were monitoring. I don't see that we are going to do this in the near future, but maybe in the future it could be, something that it will come out. Right now, as you all see, this is a new technology. People is starting to get more familiar with all of this. So it's going to take a few years before we can see the implementation.
Awesome. Thank you so much.
Thank you, Ashley.
Thanks,
Okay,
Ashley.
thank you all.
Cass, you had asked if the report is going to be reported out. So the report is already on a website. I just copied that link again. But we can talk with Portland Harbor Community Coalition about other ways they may want to publicize it. Well, thank you all so much.
Good to see you.
You too. Thank you so much.
Thank you so much, Laura. Take care. Bye.
You too. Bye-bye.